Welcome to Chapter 5, Microbial Metabolism. In this chapter I'm going to split it into three parts. There's an awful lot of chemistry, a lot of heavy thinking, so we're going to separate it by three different groups within the chapter. This chapter fulfills three objectives. From Objective 2, mainly, learning about microbial metabolism and growth. From Chapter 3, this will help you understand the theory behind culturing microbes, including the use of different types of media, incubation temperatures, pH, etc. And then for Objective number 4, this chapter will help you understand the interactions and impact of microorganisms on humans and the environment. In Part 1, we're going to discuss catabolic and anabolic reactions, enzymes, and energy production. In Part 2, we're going to talk about carbohydrate catabolism, lipid and protein catabolism, and the biochemical tests that we can use to help with bacterial identification. In Part 3, we're going to discuss photosynthesis, metabolic diversity, biosynthesis, and integration of metabolism. To begin this chapter, we're going to talk about how catabolic and anabolic reactions relate. Now you're going to have to remember back to the infamous Chapter 2, macromolecules and different reactions, and we're going to talk about catabolic. You'll remember catabolic is where you break down a large molecule into smaller molecules, releasing energy. Anabolic is putting two smaller molecules together, and it takes energy to do this. So here's what happens. You take simple molecules such as glucose, amino acids, glycerol, fatty acids, and through anabolic reactions you produce complex molecules. Now you got to have energy. ATP, we're going to be talking about ATP a lot because this is the energy currency of the cell. ATP donates energy to these anabolic reactions. Remember, we've got to put energy in. Now you always lose some energy as heat. That's why you have to continue eating. It would be nice if we just got one big wallop of energy and it just kept right on going for the rest of our lives, but that's not how it works. You always lose some as heat. So ATP comes in and donates energy by a catabolic reaction where one of the phosphate groups is clipped off and you get ADP. Okay. So then we use these complex molecules either as functioning within the cell, structure of the cell, storage of energy. When we need that energy, we undergo catabolic reactions and we transfer the energy that's released by the catabolic reactions to make more ATP. So we take ADP and a phosphate group and we make ATP. And you lose some from heat, you're always going to lose a little bit of energy, and you end up with simple molecules again. So it keeps going around and around and around, where you use ATP to make bigger molecules, and you break down bigger molecules to make ATP. Here's an analogy that works for me. I work to produce money. That money goes into my checking account. Okay, I've expended energy, I've worked really hard, I've sweated, or sweat, I'm not sure. And then I use this energy to pay for things that I want. Food, mortgage, rent for some of us, paying for clothes, education. And that empties out my savings account. So I've got to go back to work producing more money for my checking account, more ATP. The cell does the same thing. Brings in complex molecules, breaks them down to make ATP. Then we take simple molecules, use ATP to put them together to make the molecules that we need for the structure and function of the cell. Now we're going to talk about how enzymes help in this catabolic-anabolic process. First off, we're going to talk about activation energy. We're going to talk about what a catalyst is. We're going to talk about enzyme specificity and efficiency. We're going to go through that part really fast, as well as going through the naming of enzymes really fast. We're going to talk about the mechanism of enzymatic function, factors influencing enzymatic activity, and inhibition. Under the category of activation energy, remember that molecules and atoms are constantly moving. The atoms within a molecule are vibrating, they're moving around, the molecule itself is vibrating and moving around. Okay, now in the book it talks about collision energy, this is pretty much the same as activation energy. Basically, okay, I have my reactant, okay, and I'm going to undergo a catabolic reaction. I'm going to take my reactant and I'm going to make two products and I'm going to release this amount of energy. But it takes energy to break those covalent bonds. Now, and we call this activation energy or the energy of activation. Okay, the book calls it collision energy. So without an enzyme, you need this much energy. But if you have an enzyme, you only need this much energy, and it happens more easily with less energy expenditure, and it's also controlled. Here's an example. Given enough time, a piece of wood will break down into carbon. It may take thousands of years, but eventually it's going to go and break down into carbon. But if I add energy to it, if you remember the scene from Castaway when he takes the stick and he's rubbing it back and forth against the other stick, and he really has to work at it and really has to work at it, and eventually it bursts into flame. And he has fire and he's all happy. Consider that to be breaking down the carbon in the wood by without the reaction of the enzyme. Okay, takes a long time, takes a lot of energy, takes a lot of heat from the friction. But if he'd had a match, think of that as the head of the match being the enzyme. Strike the match, flame bursts up in a controlled manner quickly, and he gets his fire going. Unfortunately, he didn't have any matches, but you have matches for reactions within your cell. We call them enzymes. Now, different enzymes react to do their job at a different rate. Okay, when conditions are perfect, the reaction rate is very fast, and it's as fast as it can go. When conditions aren't perfect, then the enzyme works slower. Consider it this way. If you have a class at 7 o'clock in the morning, maybe your reaction rate is going to be a whole lot slower than if you have to get to a class at 10. Unless you're a morning person, and then it's the exact opposite. Now let's talk about enzymes in general. Their function as catalysts, their specificity, efficiency, and naming. A catalyst is a molecule that's involved in a reaction that doesn't undergo change itself. That's a catalyst. You're all familiar with the term catalytic converter. In your cars, it helps convert some of the pollutants coming out of the exhaust into things that are less polluting, but the platinum that is in the catalytic converter itself is not changed. Okay. Enzymes are protein catalysts. They are proteins that engage in a reaction without being consumed or changed by the reaction themselves. Okay. And they do this by lowering the activation energy, like we talked about in the previous slide. Now, enzymes, generally speaking, are very specific for the job that they do. I call this a wrench versus a hammer. If I want to tighten a screw, am I going to use a hammer? No, only if I only have a hammer. If I want to tighten a screw, I'm going to get a wrench. Now, on the other hand, you can hammer in a nail with a wrench, but it's a whole lot easier if you use a hammer. Enzymes are very similar. We have enzymes because of their shape are suited for a specific job. Generally speaking, they do one thing and they do it very, very well. We're going to talk more about what they do as we go through this chapter. They're very efficient because they are the right shape and they do just one job, then they're very efficient at that job. Now on to naming. Basically, if you see an ACE on the end, it's an enzyme. There are a few that don't, but let's take an example. Okay, I've got an enzyme that will take glucose and fructose, remove water, and hook it together to make sucrose. Okay. It's very efficient in doing that. Okay, and we're going to name it according to what it does. Now let's look at the other way. Now if I take sucrose and I have an enzyme that's specific for breaking it, Okay, it's not going to be the same enzyme that puts glucose and fructose together to make sucrose, because your cell wants to control these different pathways. So we have different enzymes for doing the different things. So the enzyme that takes sucrose, breaks it down into glucose and fructose, is called sucrase. It's got sucrose in the name. We know part of what it does. Now there are a few enzymes that don't have ACE in the name. For example, papain. It's a digestive enzyme, and it's found in papaya, which is why we call it papain. It's in the papaya to help the baby papaya plants digest different things within the fruit as they're growing, and you'll find them in health food stores to help in human digestion. Let's talk about how enzymes actually do this. Okay, I've got an enzyme here. This is a model. This is what we actually think some of the enzymes look like. And they have an active site, a little dimple or a groove that the substrate, that which the enzyme is going to work on, goes into the active site, and it fits perfectly. Then the enzyme does whatever it's going to do to the substrate. In this case, it's going to break it. This is a hydrolytic enzyme. The products are spat out, and the enzyme is ready to go again. What we think happens at this point is the binding of the substrate to the active site causes a change in the shape of the enzyme. Call it a conformational change, a change in the shape of the enzyme. You can see it better in this diagram down here. When the substrate goes into the active site, the enzyme folds around the substrate. We think that this conformational change produces stress on the covalent bonds of the substrate and causes it to break. Now, if this enzyme is an anabolic enzyme and putting things together, it'll change shape, force the two substrates together, and kind of like an arranged marriage, and they get together and form one bigger molecule. Now, you'll notice on this diagram we have another dimple. This is not the active site. We call this an allosteric site, and it's important in regulation of the enzyme, whether you turn it on or turn it off. We're going to talk more about that in a few slides. Enzymes, as well as being very efficient in what they do, are also very, very picky. Different enzymes like different conditions to work at their optimum speed, their optimum reaction rate. Let's talk about temperature first. What would you, if you had to guess, what would you think would be the optimum temperature of the enzymes that are functioning within your body right now? Body temperature, 37 degrees. If you lower that temperature too much, they start slowing down, and if you raise it too much, then they start coming apart. Remember denaturing from chapter 2? If the temperature gets too high for what the enzyme wants to do, for what is good for that particular enzyme, it'll start vibrating too much. Bonds that are holding it together into its tertiary and quaternary structures come apart, and it unravels into its primary structure. Okay, and then when it comes together, it forms a blob, a non-functioning, non-enzymatic protein. Okay. Now, don't think that all enzymes like to function at body temperature. We have isolated some bacteria from the deep ocean. In fact, they've been living off of sunken well carcasses, living off of the blubber. Yum. And we pull these organisms out, and their enzymes like to function at very cold water temperatures, about 4 degrees C. You know what we use those enzymes for? Breaking up stains in laundry detergent. We put them in laundry detergent, they break up stains in your washing machine, even at the cold temperatures that make it more economical for us. Okay. There's also bacteria that we've isolated from hot springs in Yellowstone that like to live at close to boiling. Some of them like to live at even higher temperatures than boiling. It's very interesting that there are enzymes that can function at a wide range of temperatures, but each individual enzyme has a temperature that it operates best at. It's the same thing for pH. Going back to those hot springs in Yellowstone, most of them are very acidic. They have a very low pH. The enzymes will not function at a neutral pH. They just need those extra hydrogen ions to maintain the shape and function of the enzyme. Now, taking an example close to home, you have an enzyme called pepsin that is produced by your stomach. It operates best at a pH of about 2 to 3. Now, when pepsin exits your stomach into your intestines, your liver produces bile salts. They're concentrated in the gallbladder. They're squirted into your intestine, and it raises the pH of your stomach contents from 2 to closer to neutral. Pepsin stops working because it doesn't like that pH. That's actually a good thing because pepsin breaks down proteins, and you really don't want the proteins in your intestines, say your intestinal cell walls, getting broken down. Other enzymes kick in that work better at a neutral pH. Okay. And substrate concentration, that's the last thing that we're going to talk about on the category of enzyme activity. Enzymes will work slower when there's less substrate. Makes sense. They're sitting around waiting for an enzyme they're sitting around waiting for a substrate to come bumping into their active site. When you have more substrate, you keep getting faster and faster enzyme activity until it's working as fast as it possibly can. And giving it more substrate is not going to make it work any faster. I'm sure all of you have had work situations like this, where you're working at your peak and giving you more work is not going to make you work any faster. You don't want enzymes working 100% of the time. It's kind of like leaving the lights on in your house, except for in this situation, if you leave the lights on your house, the bulbs get hotter and hotter, and eventually you burn down your house. You want to be able to turn enzymes on and off just like you turn on and off the lights in your house. We call this inhibition of the enzymes. So here's the normal situation. You have the enzyme, it's got its active site, the substrate comes in, fits perfectly, the enzyme acts on the substrate, produces the products. Now, there are some inhibitory substances that are either, in this case, the same shape as part of the active site, but not all of it. It's not the substrate, but it fits in, but it doesn't fit in perfectly. This is called competitive inhibition. It's competing with the substrate for the active site. So the enzyme says, hey, wait a minute, you're not the substrate. Puh, spits it out, then the substrate can come in. But this slows down the activity of the enzyme. Your cells do this. There are also some poisons that do this, that go in and bind to the active site irreversibly. Your cells, your enzymes are not able to spit it out. Okay, that's why an awful lot of mushrooms are poisonous. They have non-reversible competitive inhibitors. You don't want to eat them. Okay, next, here's this allosteric site. Remember the allosteric site? This little dimple that is not the active site. You will have a regulatory enzyme that will bind to that allosteric site and change the shape of the enzyme so that the active site doesn't fit the substrate. The substrate bounces into it, doesn't fit, goes off without being changed. It's kind of like flipping the electricity off so that your light bulbs aren't on. Now, here's an allosteric inhibition where you have a regulatory element that fits into the allosteric site that we call feedback inhibition. Say there's a process where we use several enzymes to accomplish one goal. Say we've got amino acid A and we need more of amino acid B, but you can't take amino acid A and just immediately make it into amino acid B. You've got to change it to shape like this, you got to change it so it looks like this, and then at the end you get this. So we have 1, 2, 3 enzymes that are involved in this process. So enzyme A does this job, enzyme B does this job, enzyme C does the final job. So what happens is in feedback inhibition is the product of the end of the pathway will go and bind to the allosteric site of the first enzyme, turning it off. So here's the practical application. When I get enough of amino acid B, it turns off the process at the start. So here's an analogy. I'm making bicycles, okay, in my bicycle factory, and I run out of the bolts that I need to put wheels on. Okay, I don't want to stop the manufacturer halfway through and store a whole bunch of half-made bicycles. I want to stop it at the very beginning. So I either have a bunch of bicycle parts that are easy to store or a whole bunch of bicycles that I can sell. Okay. Now once I get a shipment in of the bolts for putting the wheels on, I can start the factory back up again. Okay. It's a similar process where the final product shuts down the beginning of the pathway. We call that feedback inhibition. On to energy production, which is the last section for part 1. We're going to talk about redox reactions, generation of ATP, and metabolic pathways. Part of energy production involves redox reactions or oxidation reduction reactions. So here's an example. Molecule A takes an electron and donates it to molecule B. In this process, A is oxidized, B is reduced. Energy has been transferred with the transfer of the electron. This can also happen when you transfer a hydrogen. Remember, a hydrogen is an electron and a proton, so sometimes the proton comes along for the ride with the electron. In the process, you get passing of energy from one molecule to another. So here's a good way to remember the difference between oxidation and reduction. My favorite is Leo goes grr. Lose an electron, it's oxidation. Okay, so A has lost an electron, so it's oxidized. Grr, gain an electron, you're reduced. So B has been reduced. Now a lot of students prefer oil rig. Oxidation is losing an electron, reduction is gaining an electron. You're probably wondering, why in the world do you have to remember this? In part 2, we're going to be talking about a lot of redox reactions, and you'll get to see some actual examples. The thing to remember is the reduced molecules have the energy. They have more energy than oxidized molecules. In other words, the one that has the electron has the energy. Let's talk about another type of energy transfer. When you move phosphate groups around, it's kind of like moving that electron. You move energy with these phosphates. So we call this phosphorylation, when you put a phosphate group onto a molecule. Now in part 2, we're going to be talking about substrate level phosphorylation and oxidative phosphorylation. Let's talk about what we mean by that. Substrate level phosphorylation is where I move a phosphate group from one molecule to another one. So if I take this phosphate off of ATP and I put it onto, say, glucose, I have phosphorylated the glucose. The glucose now has more energy than the ADP. Okay, ATP has more energy than ADP because it has one more phosphate group. Now, oxidative phosphorylation, we're going to talk about that during the electron transport chain. That's where you take a phosphate group, an inorganic phosphate, that's just floating around, it's not attached to a molecule, and we slap it onto ADP and make ATP. So as we go through the process, I want you to keep track of which one is substrate level phosphorylation and which one's oxidative phosphorylation. In part 3, we're going to talk about photophosphorylation. It's very similar to oxidative phosphorylation, but we use light to provide the energy to put the phosphate onto ADP to make ATP. To finish up this section, let's go back and talk again about how ATP enables us to perform anabolic reactions and how we generate ATP from catabolic reactions. Okay, so here I've got substrate A, and I take an ATP, I take a phosphate group off of it, and I put it onto B. B has been phosphorylated through substrate level phosphorylation. Okay. Now, later on, if I take the phosphate group off of B and I transfer it back to ADP to make ATP, then ADP has been phosphorylated. This is where we get the energy to do things. Generally speaking, if you phosphorylate, okay, if you're taking the phosphate group and you're putting it on to a molecule, that's an energy requiring reaction. Okay, it's endergonic. If I'm taking it off, that is an energy releasing reaction. It's an exergonic reaction. Now you'll notice that an endergonic reaction is happening at the same time as an exergonic reaction. Removing the phosphate from ATP is exergonic. I release energy here and transfer it to here in an energy requiring reaction. Here I release the energy, okay, from B, and I put it on, and that's an energy requiring reaction. So we go around and around and around, moving the phosphate groups around to produce and release the energy, okay, for the different reactions. Redox reactions are very similar, okay, but in this picture we're using that the phosphate as an example of this type of thing. Whew, that was a lot of material, wasn't it? Go ahead and take a break, stand up, move your arms around, get the blood flowing to your brain. If you don't get it the first time through, do not despair. They've found that for most of us, if it's something completely new, if it's a completely new idea that you've never heard before, it takes exposure to it about 6 times. If it's something that you are familiar with, okay, then it takes fewer times of being exposed to it to remember the material. So don't despair. Remember, repetition, repetition, repetition. So go back, reread the text, rewatch this portion of the PowerPoint presentation as many times as you need to.